In all areas of life, cooperation can be found on many levels. Why does cooperation evolve in nature? How does cooperation evolve? Why would cooperation evolve if it would be easier to take without giving back?
What does cooperation really mean? In its simplest form, it can involve two things working in harmony, while in its complicated forms, it can involve complex workings like the human body, relationships, even the social patterns of a nation. But what about in the simplest form of them all, bacteria? Can cooperation really occur? And if so, how? Using methylobacteria and E. coli as a model, we will be exploring this very interaction. Both E. coli and methylobacteria need a carbon and nitrogen source to survive. So E. coli uses glucose as a carbon source, and methylobacteria uses methylamine as a nitrogen source. But E. coli cannot use methylamine, and methylobacteria cannot use glucose. It is so strange that when they're grown together in a media of just glucose as a carbon source and methylamine as a nitrogen source, that they somehow survive. We think that one is making the food for the other. Actually, let's go to the lab to find out.
An easy way to study our consortia is to measure how much of each food nutrient source, carbon and nitrogen, the bacteria use as they grow. So we took these two bacteria, E. coli and methylobacterium, we grew them up, and then we measured how much of them grew and different concentrations of food source. We wanted to ask ourselves if the bacteria would grow better while we increased the concentrations of food source of one of the nutrients, but held the other one constant. Essentially, with this data, we could calculate the carbon to nitrogen ratio of the bacteria growing in consortia. We made a graph with the kind of data we expect. We held our nitrogen source, methylamine, constant at a concentration of 1.25 millimolar. We then varied our carbon source, glucose, at many concentrations. Then, we measured total growth of the consortia growing under each condition. The potential growth is expected to plateau once they are constrained by the limiting resource, nitrogen.
Okay, so in our experiment, we grew many different types of E. coli with an ancestor methylobacteria. And each of these E. coli mutants, let's see here. Okay. So each of these E. coli mutants have a gene knocked out, and they're all different, and we have all different types here. And we have an ancestor in here as well. And basically, a gene is knocked out means that it's no longer working. Right, so if we analyze the growth patterns of these when we grow the different E. coli mutants with the methylobacteria, we can see if it grows better or grows worse, that we knocked out something very important.
Ideally, we would know everything that these bacteria are making and when. With this kind of data, we'd be able to see how fast they eat glucose, what products are made, and when they are eaten. Our experiment involves screening for a significant set of compounds in the material that these bacteria grow in.
We grew up 12 different flasks of our cooperating bacteria and measured them at different time points. With the media we collected, we could run a host of assays to find out what is going on in each of these flasks. In everyday life, we find cooperation in cities, countries, everywhere. People working together to create a system greater than the sum of its parts.
Okay, it's a wrap, guys. Great job.